We describe a stencil-based, lithography-free, single-cell micropattern method that overcomes many obstacles associated with photolithography and soft lithography. The stencils are reusable and process is inexpensive and customizable.
This method opens a new avenue for non-engineer researchers to study cell shape and area-induced mechanotransduction in various physiological and pathological contexts. Furthermore, this protocol allows for micropatterning of thousands of cells, which can be utilized for high-throughput drug screening and disease modeling in a dish.
Visual demonstration will facilitate the implementation of two crucial steps. Detach one cover slip from hydrogel, drying hydrogel sufficiently before placing the stencil.
Prepare the polyacrylamide hydrogel solution in a new 50 ml polypropylene centrifuge tube as described in the manuscript. Next, to prepare the 5% photoinitiator solution, dissolve the ingredients in 700 microliters of 100% ethanol. The ingredients are not water-soluble, so be sure to fully dissolve by vortexing. Add 300 microliters of PBS for a final volume of 1 milliliter. Working on ice, dilute basement membrane matrix protein stock solution with ice-cold DMEM F12 media or 1x PBS to the optimized dilution ratio. Keep on ice for later use. When working with basement membrane matrix, make sure to always work on ice and use ice-cold solutions in chilled pipette tips and tubes. Place a UV bench lamp in a biological safety hood. Next, place the sterilized paraffin films prepared previously.